We're going to take a look at antibiotics. The technical term for an antibiotic is a chemotherapeutic agent, an antimicrobial chemotherapeutic agent. That is right. Antibiotics are chemotherapy. We just don't call them that because people get freaked out when they hear the word chemotherapy. Anyhow, we're going to take a look at classes of drugs, their mechanism of action, and the mechanism of resistance, and then finally we're going to wrap up with the clinical decision making on how you choose the right antibiotics. Okay, so antibiotics, broad look, will have one of a few major mechanisms of action. They'll either inhibit the cell wall, inhibit or damage the cell membrane, prevent protein translation at the ribosome, either at the 50S or 30S subunit or both. It will inhibit a pathway for the biochemistry of some needed substrate, or it will mess with the RNA or DNA. The big group of cell wall inhibitors are listed here. So penicillin, cephalosporin, carbapenems, and monobactams, these are all beta-lactams as a group. All of these inhibit the cell wall in pretty much the same way by inhibiting an enzyme known as transpeptidase. Sometimes it's known as carboxytranspeptidase or dialyltranspeptidase, and sometimes it's called penicillin binding protein because this is the protein that was discovered whenever people were trying to investigate penicillin to see how it worked. This is the enzyme that was discovered that penicillin binds to. Vancomycin, on the other hand, binds to penicillin's substrate, which is diala-diala, and I'll explain what that means in a minute. So you can either bind to and inhibit the penicillin binding protein. You can inhibit this penicillin binding protein substrate by binding to the substrate. And there's a few other mechanisms of action for inhibiting cell wall. For example, bacitracin actually inhibits a different aspect of cell wall synthesis. So what happens is inside the cytoplasm of the bacteria, it puts together the monomer subunits of the cell wall. And I'll show you what those are in a second. But it puts those together in here, and then the bacteria will transport those subunits across and add them to the growing cell wall. The carrier molecule is known as isoprenyl pyrophosphate, and so without isoprenyl pyrophosphate working, you can't carry the cell wall subunits across the membrane to connect them. So here's the basic makeup of a bacterial cell wall, and the reason I'm pointing this out is because the cell wall of a bacteria is the most unique thing about bacteria, and it's the thing that makes it an easy target for antibiotics, because if a human doesn't have a cell wall, and you make something that only destroys cell walls of this type anyhow, then you're only going to hurt bacteria. You won't have any toxicity, theoretically, on a human or on the person you're treating. So what we have are two different kinds of sugars. So the N-acetylmuramic acid, or NAM, as it's called, and N-acetylglucosamine, or NAG. And so what the cell wall is said to be made of are these NAM-NAG, NAG-NAM repeating subunits of sugars. And so they're connected together, but then also these NAMs are cross-linked by a peptide chain. So basically what we're looking at here is one sheet of the cell wall. Now, this sheet will be stacked on top of another sheet of the exact same thing, which will be stacked on top of another sheet. Gram-positive organisms have several sheets of these sugar polymers stacked on top of each other to make their cell wall. So let's take a really close look at the exact cross-linking, and I'm going to zoom in for this. We come over here, and what we see is that hanging off of every one of these NAM sugars is a string of 4 amino acids: L-alanine, D-glutamate, L-lysine, D-alanine. And then if we come back down here and look, this is a string of 5 peptides. This is called a pentaglycine chain because it's got a penta meaning 5. It's 5 glycines strung together. Now you only see this pentaglycine chain in gram-positives typically. In gram-negatives, what happens, instead of using a pentaglycine chain to connect these, this peptide chain to this peptide chain, and gram-negatives, they do a direct linkage instead of using a glycine chain. So without those chains, those cross connectivities, then you don't get the cell wall. And so over here I've got the synthesis of the cell wall written out, and basically what it boils down to is 4 basic steps. Each of these steps could be expanded on. For example, step 1 is really 7 steps, but you form monomers in the cytosol, and like I said, 7-step process for that. Then you use a transporter, and this is the isoprenyl pyrophosphate. It's also called bactoprenol. And then so you transport it out of the membrane, and then you add the monomers to the growing polymer, and then finally you form the cross linkage using the diala. And again, I'll show you what I mean by that. So right here I have the monomer. This is what's made inside of the bacteria cytoplasm and shipped out. And what you'll notice here is I have 5 amino acids on here and only 4 amino acids in the end. And what happens, this is 2 dealanines together, and usually it's abbreviated diala. And what happens is the transpeptidase, let's say that here's my glycine that's getting ready to be attached to it. Transpeptidase, my pentaglycine, transpeptidase will take this, attach it to here, and then kick this thing off. So what you end up with is an additional dealanine floating around outside of the cell. And of course, if this was a gram-negative, it wouldn't be making the linkage between a pentaglycine. It would be making it with the L-lysine of the other chain. Now I want to let you know that some sources, some textbooks, are going to say that this diala gets connected to diaminopimelic acid. Basically, this is a derivative of lysine. So saying lysine, I'm saying diala gets connected to L-lysine, which is basically mesodiaminopimelic acid. Sometimes it's MDPA, and just depends on what source you read and what it's going to be called. So why do I make all this fuss about this diala? The reason is because, first of all, it sounds like a rap song. Diala. Anyhow, the second reason is because vancomycin binds here and prevents transpeptidase from acting on it. The next group of things inhibit or interrupt the cell wall. So the polymyxins and the diaptomycins, they will interrupt the cell wall. And now the reason this, so these aren't used a whole lot because every creature has a cell, not a cell wall, it interrupts the cell membrane, But every animal, and we're animals, every animal has a cell membrane. And so the bacterial cell membrane is different enough from ours that we can target things in the membrane and it'll be more toxic to the bacteria than it is to us. However, these things are still pretty toxic. They can damage especially the kidneys, and so their use is pretty limited, but they're still useful for some antimicrobial therapies. Now, there are a lot of things that target the DNA and RNA of bacteria because bacteria use different enzymes for their polymerizations and for their gyrase and everything else. They use a lot of different enzymes than humans do, and they're so different that they're easy to target. For example, the quinolones, these inhibit gyrase. Rifampin inhibits RNA polymerase. And there's just a lot of different ways to target the bacteria DNA. First of all, bacterial DNA is in a circle, it's in a loop. And so whenever it finishes transcribing, it actually makes a loop inside, like 2 rings hooked together, and then it has to unhook these. And you can target that action and prevent the unhooking of the DNA, and that will prevent it from dividing. Some antiviral agents will actually they'll use the fact that the enzyme for the polymerization is different enough that they can add in basically fake nucleotides and stop DNA transcription altogether whenever it comes to viral DNA and RNA. Next, we can inhibit metabolic pathways, which is what I'm talking about right here. So preventing substrates to going to products. This is especially useful if those substrates are necessary for the cell's life. And so that's what sulfa drugs do. And sulfa and trimethoprim, they'll inhibit folic acid synthesis. Now, Folic acid. I know you've heard of it. I know you know it's good for you, right? Everyone has to take some folic acid. Really good for mothers to take before they get pregnant. All these good things about folic acid. So how is it that we can inhibit folic acid in bacteria and not really mess with humans? The answer to that is we get all of our folic acid from our diet, while bacteria make all of their folic acid from a substrate called PABA, para-aminobutyric acid. And so sulfa drugs will act at, there's several steps to getting this to folic acid, several different steps. And so sulfa drugs and trimethoprim both act at 2 different steps in the pathway. And so these drugs are usually you'll use trimethoprim with a sulfa drug called sulfamethoxazole. And whenever they're used together, you can actually use a lower dose of the 2 because they work synergistically. And that way you do it's less toxic to a person. It hurts the person less taking it. There's a lower toxicity and there's a higher antimicrobial activity. Now, the ribosome of bacteria are so different from humans that we can target their ribosome and prevent them from making basically any proteins at all. And you know, if you don't make any proteins, you die. And so that's the idea here. Instead of, like, if we were to just inhibit one protein, yeah, that's great. Like we inhibit transpeptidase or we inhibit dihydrofolate reductase to prevent folic synthesis. But here we can just stop all proteins from being made and kill the bacteria that way. So you have some things that will bind to the 50S ribosomal subunit, some to the 30S, and some will linezolid will bind to both the 30 and 60 and prevent that from happening. Now the thing with the ribosomes is that we have mitochondria inside of all of our cells, and our mitochondria have their very own ribosomes. And the ribosomes of a mitochondria are very similar to the ribosomes of a bacteria. And so using these medications will have an effect on the mitochondria. And so you can actually be toxic to your own cell's mitochondria. And so that's kind of limits the therapeutic index on some of these. It makes them more toxic than what they otherwise would have been. Okay, so let's look at mechanisms of drug resistance. So some bacteria become resistant to the antimicrobial agents that we are giving them. Now, before I start, I want to point out that I didn't point this out earlier and I intended to. This is what's called a beta-lactam. And I said we have 4 different things that are in the beta-lactam family, including penicillins, cephalosporins, that kind of thing. And so this is a beta-lactam. It's a lactam. A lactam is basically an amide ring with the carbonyl group. But whenever that nitrogen is in the beta position, it's called a beta-lactam. And all beta-lactams, at least that I know of, at least all the beta-lactams that are medically important are a 4-sided ring. And so one way that drugs become resistant is by inactivation, and this is exemplified through the beta-lactams. For example, a lactamase will actually break this bond right here, and then it will add, it breaks the bond by adding water. So it'll add an OH group here, and then it'll add a hydrogen here, and breaking this bond at the same time. So it breaks the bond of the beta-lactam, and that inactivates the beta-lactam, makes it not work anymore. One way we've learned to get around that, in some infections, we can actually add a lactamase inhibitor to prevent the lactamase from breaking open the beta-lactam so that the beta-lactam can act on transpeptidase. It's like a freaking chess game. The other thing they do, we're looking at big strategies here. Inactivation, the next one's permeability. So some of the drugs will use different pores to get into the bacteria. If the bacteria have a mutation or something, the shape of that pore changes and the drug can no longer get in, and so it becomes resistant. With that same thought in mind, we could look at efflux pumps. Maybe the drug can get in, but they're just being pumped right back out. So whenever a microbe gets the genes for an efflux pump and it starts pumping out all of the drugs that we're putting in it, then it no longer is harmed by those drugs. They can change the binding site where the drug binds to a protein. This can happen to any kind of protein. Right here, the example is the ribosome. And let's say that the ribosome, typically we bind to the 50S subunit right here. Well, if the shape where that binding site was changes, then our drug can no longer bind to it. And so it is now resistant to that drug. Lastly, the real big strategy is alternate metabolism. This is really seen in sulfa type of things, and so if you can't use the normal pathway to make your folic acid, then you find a different pathway, because if the drug is blocking this, you just go down this way and you get to your product a different way. There are a few other mechanisms that they're not specifically called mechanisms of drug resistance, but they do actually confer drug resistance. The first is called a phenotypic change. If you change your phenotype, if you change the way you look, your morphology, then you can become resistant to a drug. And one of the things is bacterial cells will convert to an L form in some cases. An L form, so this is where a gram-positive bacteria says, you know what, I don't need my cell wall anymore, I'm going to live without it. And so usually without the cell wall, they become osmotically lysed, but they have figured out a way to make themselves isoosmotic or whatever so that they can turn into this L form and live without their cell wall. The other thing is biofilms. So biofilms do a couple of things. First of all, they prevent the drugs from penetrating, which back in the 80s, early 90s, we thought that that's the only thing the biofilm did. We thought, oh, biofilm, it's just stopping the drug from penetrating, and that's why it's not killing the bacteria. But that's not completely true. In biofilms, bacteria become quorum, and what that means is they start communicating with each other, and they start sharing information, and they start changing their morphology quite significantly. And so the morphology and the phenotype of a bacteria inside of a biofilm will be different than those that are free living. And one of the new strategies that's kind of working towards attacking this is that we're working on strategies to attack the quorum sensing molecules, the quorum, the messages going back and forth between the different bacteria, so that they no longer realize that they're in a biofilm, so that they no longer realize that they're in this big community, and they change their phenotype back, and then we can kill them again with our normal drugs. Another thing would be resting state. So if a bacteria is in a resting state, meaning its metabolism is almost next to nothing and it's just being inactive, it's not dividing, then we can pump drugs all day long and it's not going to do anything to the bacteria. And then we think, oh, the bacterial infection's gone, we quit giving drugs, and the bacteria all of a sudden they're like, hey, I think I want to come back to life again. I think I want to start doing things. And so that will be a mechanism of drug resistance, and especially some bacteria that seem like they can actually sense when the antibiotics are being given, and at that very point they go into a resting state, and then whenever the antibiotics are gone, they wake right back up. So how do we treat bacteria that have become resistant? So the first thing is we find out what the microbe is susceptible to. Every microbe is going to be at least susceptible to one drug or another, and there are only very, very few exceptions. There are 3 basic tests to find out what a microbe is susceptible to. The very earliest of these, and probably not used at all anymore, is called the Kirby Bower disc diffusion test. We're going to go over it just so you have a little historical background. But then there's also the E-test, which is semi-rapid, and then there's the tube dilution test, which takes a little bit more time, but it's the most accurate. So when you find out what the bacteria is susceptible to, the key to treatment is using the narrowest spectrum antimicrobial that you possibly can. And the reason for that is it will prevent bacteria from becoming resistant to our more useful medications. Anyhow, here's the Kirby Bower disc diffusion test. I'm going to zoom in up here first. So what you do is, here we have a bunch of different discs, and each of these discs are impregnated with different antibiotics, with different antimicrobial therapeutic agents. And so you plate this Kirby Bower plate with bacteria, and before it starts growing, you place these discs in different places on the plate, and you let it all grow. When the microbes grow, they can't grow near the antibiotics because the antibiotics are inhibiting their growth. And so then you can measure across the diameter of this zone of inhibition, and then you compare that zone of inhibition to a chart. Now these charts were made through regression analysis, and so they're not perfect, and they're not going to be exactly specific for your microbe, but they're going to be very close. The reason you need a chart is because, well, let's go back and look. Let's say that this is one antibiotic, and let's say that it actually diffuses out this far because it's a really small molecule. Of course, it's going to be in low concentrations out this far, but let's say it diffused out this far, and you're only actually inhibiting the bacteria at the extremely highest concentration. Let's take example this one. Let's say that it was plated here, but it only diffuses this far because it's a larger molecule, it doesn't diffuse very well. Well, you're actually inhibiting the bacteria for most of the concentration. And so in either case, or we could say the other way around, we could say that this one right here actually diffuses all the way across the plate, but then even though this circle is wider than this circle, it won't be actually as impressive if this one only diffuses like that far. So that's why regression analysis was used to come up with charts like this. And you can come over here and say, okay, let's see, amoxicillin, am I using it for any other bacteria, or am I using it against staph? If I'm using it against staph, then it doesn't become susceptible until the zone of inhibition is 20 or more. But if it's any other bacteria, then it becomes susceptible if the zone of inhibition is just greater than 18 millimeters. You'll also notice that there's this intermediate zone. It's just saying it's not resistant, but it's not really susceptible either. So you can kind of use some clinical reasoning on that as well. And then what this little line here is, if you purchase a Kirby Bower test set, what you're going to get is you're going to get a bunch of different discs, and each disc is going to have a different letter code on the back of it. And these letter codes are just saying, hey, this is carbapenem, this one AMC is amoxicillin, so you know what disc you're looking at. You say, okay, the CB disc is what I had here. What was it? Oh, it's carbapenem. And that's the Kirby Bower disc test. It was developed, I think, in the 50s, and it's really not a great thing for knowing exactly how susceptible something is, but it will give you a general idea. Then kind of a derivative of that is the E-test. The E-test is basically, I don't know if I can zoom in close enough to see, but what we have here is that each one of these areas is marked with the concentration of the antibiotic that's in that area. So if you come all the way down here, you can say automatically, I know that this is susceptible down to 0.125 micrograms per milliliter, because it's already got it calculated for you. And so in this case, you really don't need a chart to see what the minimum inhibitory concentration is. But again, we have to take into consideration that you don't know exactly what the diffusion is, and so maybe the concentration from here diffused down into here and mixed with this 0.125. So it's just really a rough guess. It's a close guess, but it's a rough guess. Now here's the gold standard. What we're going to do is we're going to make dilutions of an antibiotic. So here I'm going to have 0.25 micrograms. Here I'm going to have 0.12 micrograms per milliliter. Here I'm going to have half of that. And then at the very end you have no antibiotic. And so you put the same amount of bacteria in each of these. Let's say you have a bacteria that's mixed up in a broth, and you add 1 milliliter of bacteria broth to each of these test tubes, and then you wait and you see which ones grow and which ones don't. Now you're going to have several other dilutions all the way over, and I didn't make all of them in this illustration, but you're going to look all the way down to the lowest concentration of antibiotic, And whatever that concentration is, that's going to be your minimum inhibitory concentration. The MIC for this would be 0.12 micrograms per milliliter. Now what does this mean? That means that if you want to have any effectiveness in treating a bacteria, then wherever that bacteria resides, you need to have at least this concentration in order to be effective. Now that's great. Let's say you have septicemia, you have bacteria, you have bacteremia, you have bacteria in your blood. Well, then all you have to look at is what the serum concentration is. But what happens if you have, you're trying to take medication for a boil? Let's say you have some type of skin infection. Then you need to look at the concentration that it gets up to at the extracellular fluid in general. What if you're treating a urinary tract infection? Then you're going to want to look at the urine concentrations. And I'm here to tell you, I spent quite a bit of time looking for typical serum, urine, and ECF concentrations, and that information is extremely hard to find, or else I just have the wrong books. I mean, it's not on Google. I can't find it on Google. I can't find it on Bing. I can't find it on Access Medicine, Clinical Keys, and I can't find it in my 4,000-page Drugs and Information Handbook. But I digress. Let's talk about the therapeutic index, because once you know the minimum inhibitory concentration, the therapeutic index is the next thing to think about. So therapeutic index, so that's saying I want to know the toxic dose 50, the TD 50, over the effective dose 50, the ED 50. So what this means is that if we look at, we're charting the entire population and we're saying, here's the dose, and here's my toxicity. And then, so starting out, you're going to have very few people get sick, and then eventually it's going to spike up, and then everybody's going to be sick. And then 50% of the people get sick at this so here 50% of my people are sick at this concentration. So at that dose, at that concentration, 50% of people are having toxic effects. We don't want people to have toxic effects. We don't want 50% of them to have, we don't want any of them to have toxic effects. So that's why the therapeutic index is helpful. So ED50 is the effective dose. So whenever we're talking about antimicrobial agents, you can substitute the ED50 for the minimum inhibitory concentration. At what minimum dose do I get an effect? For other things where you're not treating microbiota or microbial infections, let's say, for example, you want to give somebody let's just say sildenafil. He's having erectile dysfunction. At what dose of sildenafil would 50% of your patients receive an erection? That would be the ED50. But for antibiotics, we're looking at the MIC. That's the minimum inhibitory concentration. So you can say, let's say we're going to give imaginary numbers. So let's say the toxic dose, you need 100 micrograms per kilogram of body weight. And let's say that our effective dose, our minimum inhibitory concentration, is 0.5 micrograms per kilogram of body weight, or whatever you want to do it, per milliliter. And you divide that in, and so my therapeutic index would be really high. It would be 200. This is a good thing. This means I have a lot of wiggle room whenever I'm giving this patient medicine. However, let's take another look. Let's say that if my toxic dose is 10, but my effective dose is 9. At this point, I just have my therapeutic index is just slightly greater than 1. And so the closer you get to 1, the worse that medication is. You have no wiggle room. And if you get less than 1, that means you are hurting the person way before you even get to a place where you're killing the bacteria. Another thing to think about whenever you're treating with antibiotics, if it's not on an IV drip, like a constant drip, if you're taking something by mouth or you're giving a shot, you got to realize that plasma levels go up. So you take that first dose, plasma levels go up, but start to go down over time. You take that second dose, go up, go down over time. Take that third dose, go up, go down over time. And it takes a few doses before you actually get to like this rough even kill where you want to be. And so you got to keep that in mind, that you're going to have these spikes. And does that spike go over the toxic dose? And also, whenever it comes down, does it go under the minimum inhibitory concentration? Okay, I looked everywhere. I looked online. I wanted to relate this back to our case this week. I wanted to calculate some therapeutic indexes for us, and I couldn't do that. So here's why. First of all, I found clindamycin was one of our susceptible drugs on our chart. And so I looked up to try to find the TD50. The only thing I could find was an LD50, and my source for that was Drug Facts and Comparisons 2011. It's a 4,000 freaking page book just talking about drugs and facts about those drugs. I found an LD50. It specifically stated that LD50 was measured in rats. Guess what? I can't kill a person. I can kill a rat, no problem. But I don't even want to give them a toxic dose. I don't want to give, especially not a lethal dose, so I can't really use that to calculate a therapeutic index. On the other hand, I found gentamicin's therapeutic toxic dose was 12 micrograms per milliliter in a healthy person. In our thing, the minimum inhibitory concentration was less than or equal to 0.5. So gentamicin had a therapeutic index of 24. That's not too bad. I'm saying that's not too bad because it's a number greater than 1. I really have no idea if 24 is great or not. Anyhow, a little bit more information. So distribution, it's wide distribution, goes into all the ECF on both of these. Elimination, I could find elimination information for clindamycin, 10% in the urine, 3.6% fecal. The rest of it is deactivated by enzymes. And so that gives you a rough estimate about like if you had a bladder infection or if you're treating a GI infection, what would you expect for the concentrations to be in those areas. So last of all, I want to ask you this question. How do microbes become resistant to our antibiotics? And of course I have the short answer written here, but there's a much more elaborate answer. I want to get into it. So I'll just run through these really quick first. So a bacteria can have a mutation that ends up making it resistant, or it can obtain new genes. And it can obtain new genes through transduction, transformation, or conjugation. I don't have time to explain any of those things. But just know, like a mutation, most of those are going to be harmful, and the ones that aren't harmful, most of those aren't going to confer bacterial antimicrobial resistance, antibiotic resistance, but 1 in a quadrillion, something like that, mutations will be like, bam, you're resistant to this antimicrobial agent now. So if it's really rare for the mutation to work, most resistance is obtained through this pathway, obtaining the entire gene for resistance. In order to understand this mechanism, we have to realize that almost all of our antibiotics come from microbes. So for example, the antimicrobial agent bacitracin was discovered in the bacteria Bacillus subtilis that was infecting a girl named Tracy. That's where it actually got its name, bacitracin. Of course, this way of spelling Tracy is actually not the way she actually spelled her name, but that is what researchers misspelled her name as, and that's why it's called bacitracin. So if Bacillus subtilis was producing bacitracin and it was not dying from that antimicrobial drug, then how was it not dying? The answer is bacitracin already had resistance. So every bacteria that produces an antibiotic already has resistance to that antibiotic, and it produces the antibiotic so that it can kill other bacteria and it will have a competitive advantage. Now what happens when you have this bacitracin here? He's sitting around being happy, and then all of a sudden lightning strikes and he dies. And then when he dies, all of his DNA spill out onto the ground, and then another bacteria says, "That looks yummy," and he eats it. New bacteria gets the genes that make him resistant to the antibiotic. We'll call that number 1. Number 2, we have happy bacitracin sitting here, but then he becomes not so happy anymore because he got a viral infection. And I put HIV. Bacteria don't really get HIV. I'm just saying a viral infection, and that came to mind. Then, whenever the virus gets inside of Bacillus subtilis, it accidentally mixes up with some of his genes. And so whenever the new virus comes out, we're going to call it H super V, because it has this super resistant gene. And then whenever he goes up and attacks this guy, he's going to put super genes inside of this new bacteria. Or in our last case, we have really happy bacitracin. He's a cool guy. Got his shades on at night. Got himself a little faux hawk going on. And a smile that the ladies die for. Bacitracin runs into his favorite lady, MRSA. Now, MRSA has long blonde hair. She's to die for. And she's got the eyes that make the guys go googly. And so what happens there is called conjugation. Mr. Bacillus subtilis conjugates with MRSA and shares, just says, I'm going to pump a little bit of my genome over to you. I should have made the genome white instead of blue. And now MRSA is resistant. But wait, the story's not over yet, because MRSA, she's not an only child. She has like 8 trillion sisters. These are all MRSA twins over here. And MRSA's just one girl up in the corner trying to fight for her way to get some food. MRSA has a little bit of antimicrobial resistance, but that's all she has. And the food's all the way down here. But all of a sudden, we see all these MRSAs hanging out, and we add some bacitracin, and we kill all of them. So we thought, nope, we left poor old Miss MRSA with the only access to the food. Now Miss MRSA with the antimicrobial, antibiotic-resistant gene is going to multiply like crazy, and we're going to have a lot of MRSAs with antibiotic-resistant genes. And so nature plays a big part in just conferring antimicrobial or antibiotic-resistant genes. However, we as doctors are playing an even bigger part by taking away the inhibition of the resistant bacteria. These resistant bacteria would have inhibited MRSA from growing out of control, but we killed them all without killing MRSA.